Hi everyone, and welcome back to the introductory series for the Obesity Machine Learning competition. My name is Julie Laffey, and I'm a postdoctoral fellow at the Broad Institute in the Eric and Wendy Schmidt Center. And my research there involves taking approaches from computation and from interpretable machine learning and applying this to biological contexts to understand disease. And I'm particularly interested in cancer immune contexts. So I'll be taking you through the remaining 2 lectures of this introductory series, Lecture 2 and Lecture 3. And today we'll be, in this lecture, talking about technology. So in the first lecture, you will have heard from Tiago and learn about adipose tissue and the adipocyte cells that make up this tissue, and then also how when things go wrong, this can lead to metabolic diseases like obesity. And in this lecture, we'll be talking about 2 revolutionary technologies that will allow us to both probe and profile individual adipocyte cells so that we can better understand how, as I mentioned, when they go wrong, this can lead to obesity. So we've broken down this lecture into 3 parts. In the first, we'll learn about an important concept of gene expression programs. You can think of these like the functional units that cells use to have specific functions. And then in parts B and C, we'll talk about these 2 key technologies to profile and to probe cells that I mentioned on the previous slide. So in part A, gene expression programs. So just to take a step back, and remind ourselves what a gene is. So a gene is the fundamental unit of inheritance, and our genes live inside the DNA, and so they're encoded by this 4-letter code, where the 4 letters are adenine, thymine, cytosine, and guanine, or A, T, C, and G. And these genes carry all the traits that make us up. So both physical traits like our height and our eye color and maybe our hair color, but also biological traits and often traits that are also associated with disease. And so from these genes, you can get what is a really incredible diversity of cell types and tissues in the human body. So here I'm just showing you a few examples. You can see cells in the skin, the epithelial cells, cells in the small intestine, cells that make up your blood vessels, and also the cells in adipose tissue, so the adipocyte cells that we're talking about today. And there are really 2 things that are quite incredible, I think, when we look at this. The first is just the sheer diversity of cell types that arise from our genes. And the second is the fact that every single cell has the same set of genes or the same DNA. And so how is it that this can lead to cells looking like they do in the small intestine in one case, but like they do in the adipose tissue or in the skin epithelium in another? And so the answer to this is gene regulation. And so what actually happens is that the genes which encode these traits, and as we mentioned, which are encoded in the DNA, they undergo a process called transcription, which produces RNA. And the RNA is like a copy that's made of the gene. So for every gene, you get some copies of RNA. And the key point here is that different genes are switched on in different cells. So in a cell in adipose tissue, for example, in an adipocyte cell, we would see the adipocyte gene switched on, and so we would produce a lot of RNA that corresponds to those adipocyte cells. So now we've spoken about the genes and gene regulation and how this allows certain genes to be switched on in some cells and others in other cells. And so here you can see a schematic of this. So this is an illustration of what we call an expression matrix, where every row is a different gene and every column is a different cell. And the red boxes represent when a gene is switched on or is active in a particular cell. And so what we see is that actually, rather than individual genes representing different cell types in those different tissues that we spoke about, that actually it's a group of genes that define these different identities. So for example, we have these first 3 genes here, which are switched on in epithelial cells. And so this would be maybe an epithelial gene expression program. And then we have these next 2 genes that are switched on in blood cells. So this is perhaps a blood cell gene expression program. And then finally, another 2 genes which are specific to the adipocyte cells. And so this would be an adipocyte gene expression program. And so measuring the amount of this RNA in every single cell will allow us to tell which gene expression programs are present in the different cells, and by that means really identify what these cells are and hopefully learn what they're doing. And so we spoke a little bit about gene expression programs and how these define cell identity. And here we're looking at the zoomed in version of what I showed you a couple of slides ago. This is adipose tissue. And you can see that the gene expression programs in adipose tissue create this phenotype whereby the cells are very big and they have lots of fat stored inside them, lots of lipids. And so as a function of that, they have this really thin cell membrane. And they also have the nucleus, which are these little purple specks that are pushed off to the side. So this is to permit the main function of the adipocyte cells to produce and store lipids, leading to this phenotype. But even within adipose tissue and within adipocyte cells, you actually can have different gene expressions that lead to different functions of adipocyte cells. And so in this tissue, which is also adipose tissue, we see a very different phenotype that actually represents particularly metabolically active adipocytes. And so the reason for this different phenotype is one, these cells produce a lot of mitochondria and are very mitochondrially active. And so the kind of pink tint that you see here is actually coming from the density of these mitochondria. And they also have much smaller lipid droplets, which are the white blobs, as opposed to just the very large version of those droplets that you see in the classic adipose tissue. And so if we kind of dive in, we can think of this classical adipose tissue as being made up principally of these adipocyte cells. These are actually generated via what we call a differentiation pathway from this earlier, much smaller cell called preadipocyte. And in the metabolically active tissue, this is actually specialized functions that these adipocyte cells perform. And so these are still adipocytes, but there may be adipocytes that are particularly concerned with the formation and generation of lipids, or particularly concerned with actually using up fuel and breaking it up to release heat. So we typically refer to these as differentiation programs that define these particular cell identities, and to the lipogenic and thermogenic as metabolic programs. And one reason that we're particularly interested in these programs is because of their association to obesity. And so what we know is that skewed proportions of some of these cells, in particular, for example, a high proportion of adipocytes or a high proportion of lipogenic adipocytes or a low proportion of thermogenic adipocytes is linked to obesity. And so the hope is that by really understanding these different cells, we can do a better job at treating patients with obesity. So we do know some things already. So if you remember, we introduced that gene expression programs are made up of genes that go up and down together depending on which cell they're expressed in. And so I've highlighted just a couple of the genes that we know to mark these different cells. And so you can see here, for example, PDGFRA. PDGFRA is a gene which allows the pre-adipocyte population to proliferate and divide and lead to this diversity of adipocyte cells that we can see here. And in the adipocyte cell, AdipoQ is a hallmark gene. This is a gene that allows adipose tissue to signal to other tissues and do things like regulate insulin activity. And so that's great. We already have some information and some ability to kind of identify these different populations. But really, what we would like is, first of all, to understand not just a few genes that are switched on or off in each of these different cells, but actually to be able to measure all 20,000 genes that are in the human genome and understand what their relationship is to each of these identities. The second problem here is that we know for specific genes within these programs what they're doing. Like I mentioned, PDGFRA in the pre-adipocyte and AdipoQ in the differentiated adipocyte. But what about all other 20,000 genes? So if we delete PDGFRA, the cells won't be able to proliferate as well. What about if we delete all 20,000 other genes in the human genome? Will we be able to understand better what they do?